نقود في ماس جنرال بريغهام عصراً تحويلياً في مجال الطب، حيث تعيد الأبحاث الرائدة والابتكارات غير المسبوقة تعريف معنى الممكن.
Hi, I'm Nathan Yosviak, and I'm the head of research at Mass General Brigham's Gene and Cell Therapy Institute. I oversee a number of programs that support a wide variety of research across gene and cell therapies. The Gene and Cell Therapy Institute at Mass General Brigham supports the acceleration of gene and cell therapies. We do this in the context of an extremely large system of academic labs that are in various departments and various hospitals. We bring together this community and provide resources that can be shared. Gene and cell therapies are really true genetic medicines. A lot of pioneering work in the field of genomics has identified the underlying genetic causes for a lot of diseases. What gene and cell therapies try to do is to take advantage of our understanding of that biology and permanently correct those errors. So the promise of gene and cell therapies is that they can provide a one-and-done treatment for a lot of human diseases. Intracellular signaling is a key part of the biology that underlies a lot of the treatments that we're trying to target with gene and cell therapies. One of the major advances in the last 20 years has been the ease with which we can sequence a person's and a patient's DNA. That has provided us a lot of information into the basic structure of the human genome, but has also allowed us to gather a lot of data across many different populations of people and patients. All cells have the same genetic instructions in them, but different cell types have genes turned on and turned off at different times of their development, and oftentimes what underlies the disease is a miscalibration of these genetic expressions, how much or how little a gene is expressed.
من خلال التعامل مع مصدر الاضطرابات الجينية، توفر العلاجات بالجينات والخلايا حلولاً لم تخطر على البال. يمثل العلاج بالجينات نهجاً ثورياً لعلاج الأمراض عبر إصلاح العيوب الجينية من مصدرها.
Typical gene therapy involves taking a delivery vehicle, target the cell of interest, enter the nucleus, and express a new gene, and that gene will stay there for the life of the cell. And that will be a replacement of a defective gene. Recalibrating is a way of using either gene replacement or gene editing to take a gene and either make more of the protein that underlies it or turn it off, and to do it in the cell types that you want to target. You start with the DNA in the cell. The DNA transcribes into RNA. The RNA translates into protein. This is the central dogma. How do you design the packages in a delivery system to exactly target the right cell? You can add things to the package that will only interact with the cell of interest, and you can redesign the capsids of the virals to only target the cell of interest and to get to places that where you want it to go. Not only does this make safer therapies, but it also, if you think about it, allows the costs of all this to come down. You can manufacture less, it's cheaper to produce, and you can deliver less. You don't need to produce as much of the therapy to have the same effect in the disease. RNA therapeutics have really come to the fore with the development of the vaccines for the coronavirus pandemic, and that's based on modified mRNA packaged in a lipid nanoparticle. This has been proven safe over millions and millions of doses, and one of the ways is to take that RNA molecule, and it sounds simple, but it's technically challenging, is to circularize it. If you can circularize it, that does a couple things. If it doesn't have ends, it lasts longer in the cell. So if you can then deliver that to the cell, it will make the protein that you want it to make for longer. This makes a really big deal. This means that you can develop a therapy that will make the protein for a longer duration. This opens up a wide array of new therapeutic possibilities.
العلاج بالخلايا نهج تحويلي في علاج الأمراض، يعتمد على استخدام الخلايا الحية لإصلاح الأنسجة التالفة أو استبدالها.
Cell therapies are similar in that you're targeting the actual cell of the disease. Sometimes that's replacing that cell, and other times it's taking the cell out of a patient, engineering it, and putting it back in to treat a disease. The major advancement in CAR T therapy, or chimeric antigen receptor T cell therapy, is to take those T cells and to recognize that we can actually engineer them and make them better at fighting a particular cancer. So for example, you can take a person's own T cells out of their body, you can engineer them to express an antigen that targets a protein that's only expressed on the cancer. Put those cells back into the patient. Those cells are recognized as their own cells and are not rejected. And then those T cells go and target the cancer and eliminate it. And the idea is that this can be done in a way that is safe and can eliminate the cancer permanently without recurrence. The CAR T space is really promising. It's had a lot of success in blood cancers initially, and has a lot less success in treating solid tumors. One way to do that is to engineer the T cells to target antigens that only exist on the solid tumors, but to then also secrete other proteins that can recruit other cells from the immune system to also target that solid tumor. Additionally, in the CAR T space, being able to target autoimmune diseases, to be able to design T cells in a way to turn down autoimmunity, to actually target cells that are causing damage to oneself. I'm a scientist at heart, and as a scientist, I love learning new things about the world. It's what motivates me, and I'm always learning new things. I'm always extremely impressed by my colleagues at Mass General Brigham, and every day I learn something new. I'm always thinking about how these ideas can be connected, and what really excites me is to be a part of a system that has so much expertise across so many different areas that every now and then you get to see two pieces that can be put together in a way that may not have been seen before.